All right, so we left off last class talking about glycosaminoglycans and their conformation, as well as their charge. They have this desire to be in this linear conformation, but oftentimes because they're embedded within the ECM, they're more bundled up, and the excessive negative charge oftentimes contributes to the osmotic pressure and the desire to, when there's a compressive force, to become stretched, but then wanting to go back into its into its original conformation. Here is a list of some of the glycosaminoglycans that are present within the ECM. Chondroitin sulfate is the most prevalent. Heparin is a really important one because it serves, because of its really robust negative charge, it actually serves as a depot or a reservoir for growth factors. And you'll oftentimes see in other glycoproteins of the ECM, and we'll talk about a couple later today, that they'll have heparin or heparin sulfate binding sites, and then that subsequently will be a place where growth factors can actually be bound and be present on a seemingly fibrillar ECM protein. And also noteworthy is hyaluronin, that it's the only non-sulfated glycosaminoglycan. Oftentimes there's different degrees of sulfation, and that can oftentimes, as we've talked about, the addition of a charge can alter the conformation of that protein, and that can also lead to changes in its function. And so it's just the key takeaway here is that hyaluronin is the non-sulfated GAG here, and that heparin or heparin sulfate have a role in serving as a growth factor depot. So as we talked about, there's a class of ECM proteins called proteoglycans that, as the name implies, as I described before, usually the suffix is the one that's the most abundant here. And so proteoglycans have mostly glycans, or mostly sugars. And so to try and alleviate confusion between proteoglycan and glycoproteins, proteoglycans typically have approximately 95% carbohydrates or sugars by weight, compared to glycoproteins, which can vary depending on the core protein, but typically glycoproteins will have far less sugars. But that's not to say that the presence or addition of those sugars to glycoproteins, while they'll be much fewer, will oftentimes contribute to the function and conformation and I'll just stay with function of that glycoprotein. And some of the key functions to keep in mind of glycoproteins when they're glycosylated, or when they have those sugars added, is it'll alter, and oftentimes that alteration is improving their ability to bind to growth factors, to bind to integrins. So proteoglycans can vary in size here. An example here is decarin, which has a core protein with only one GAG chain on it, whereas aggrecan, and we talked about this in the last lecture, it's a core protein with 100 glycosaminoglycans chains on it. And so this is, and the GAGs that are on aggrecan are oftentimes chondroitin sulfate and heparin sulfate, and so this is a really important ECM proteoglycan that has a really robust role, has an important role in providing robust, promoting resistance to compressive forces in cartilage. You can kind of imagine that it almost serves like a bit of like a hydraulic role, where as the compressive force increases, like if you think about the compression on our knees and the cartilage there, as that force increases, the water swelling in for the negative charge, it'll hold it and help provide resistance to that compression because it's so glycosylated. It has so many gag chains and has such a negative charge. Some of the key functions of proteoglycans, they'll contribute to ECM assembly and organization, and they'll also be, because of the sugars that are present on some of them, for example, like aggrecan that have so many, they're able to create a porous network within the ECM, meaning that the gag chains on proteoglycans can also sometimes bind to the collagen fibers nearby, and it helps avoid just like a seemingly just like sheets of just fibers of the ECM, but rather being present within and among the collagen fibers that help create porosity. And that porosity will help provide a few important roles. It'll help mediate diffusion of different molecules through the matrix, but it'll also provide space for cells to migrate through the matrix. If our ECM was just collagen fibers, it would be pretty hard for cells to migrate through these really rigid, strong fibers. However, the presence of proteoglycans in and around collagen fibers provides space for cells to grab onto collagen fibers and actually move through. If you can imagine, this is a really crude analogy, but you can imagine it's almost like Tarzan swinging through the vines in a way where there's space for cells to actually grab onto and move or migrate through these different fibers. And oftentimes, it happens at a really slow pace. But I should have provided this. If there's actually great videos in like a 3D organoid where fibroblasts or different cells can actually push through and pull and move the matrix around and migrate through. And one last part, as I mentioned in the last slide, the gags on proteoglycans can bind to and adhere to growth factors and proteins, and they'll provide a signal during wound healing that'll mediate the migration of specific cells of where they should go and how many of them need to come in. And lastly, as I mentioned before, the proteoglycans, especially the gag chains on them, will provide a compressive strength in our cartilage. And if to try and create an illustration here, the large thick fibers are collagen fibers in the ECM there, and the looking at the box on the left, in between the collagen fibers is aggrecan, and typically when there's compression, what will end up happening is that this has such a negative charge that the osmotic pressure will help provide resistance to the compression there, and it'll have a desire for water to rush back in, and so it provides a really important role in our cartilage's ability to resist against compression. Are there any questions there? Yes. That there was a typo and that it should be aggrecan. It should be aggrecan, so that's on me. Forgive me. Yes. Yeah, so the proteoglycans that are oftentimes littered with gag chains on them have a desire, because they're so negatively charged, will want to be stretched out and have as much surface area available to the aqueous environment. However, they're space limited because of the really rigid collagen fibers around them. And so typically, whenever there's compression, especially in cartilage, if you think about someone doing really heavy squats, the compression on the cartilage will be squeezing out the water and pushing it away. But because of the osmotic pressure and the charge of aggrecan, and you can also think about how aggrecan, it's so negatively charged that when there's something that's really negatively charged all over it, and that if it's being pushed around on itself, it's going to want to repel. All of those negative charges together on that one molecule are going to want to repel back, and that helps provide resistance to the compression that's often present in our cartilage. And so the presence of that negative charge and the osmotic pressure there, it almost serves like as a hydraulic and resistance to compression. And so when the compression is removed, the water swells back in and it's able to go back to its original conformation and maintain this constant resistance to compression. Does that make sense? Are there any other questions? All right, so we talked about how proteoglycans and GAGs can provide resistance to compression. Another key part of the connective tissue is fibrillar proteins, and it's fibrillar proteins that provide resistance to tension. And the most important fibrillar protein in our connective tissue ECM that does that is collagen. Collagen is the most abundant protein in mammals. It makes up 25% of our total protein mass. So if you imagine, I don't know how many of you have made bone broth before, but it's basically like boiling down whatever chicken carcass or whatever food you have. If someone was to make a bone broth of us, 25% of that would be collagen, basically. And so as we talked about in the last lecture with collagen type 4, that all collagens, the two key features of collagen is that one, it's a triple helical structure. It's made up of 3 alpha chains that, when assembled together, make up a triple helix. I understand this can be confusing when thinking about, I know you guys are keeping all of what we've talked about in your minds at all times. Thinking about secondary structure with the alpha helix, this is not to be confused with that. This is an alpha chain that, when assembled together, makes a triple helix. And so the triple helix structure of collagen is the first key feature of collagen, the distinguishing feature. The second, as we mentioned in last class, is the GlyXY repeats. And so I mentioned that one of the amino acid residues that oftentimes is present is proline. And today we're also going to talk about the next one, which is hydroxyproline. And so what's important to note also is that it is different forms of collagen have different ratios or different degrees of proline content, but collagen is not exclusively glycine, proline, hydroxyproline. There are other amino acids that are also present. If you're interested, in your spare time, you can look up the 1600 amino acid sequence of collagen 1. And if you're so interested, you can look at and appreciate that there's glycine and oftentimes another amino acid, some other amino acid, glycine. And sometimes it'll be proline, or proline, hydroxyproline. And so if you're interested in doing that, I'd be happy to send you the link. Yes. Yes. I'm going to talk about that in the next slide. So hold that thought. And what's also important to know is that there are 42 different alpha chains that have been identified. And to date, there are 28 different kinds of collagen that have been identified. And we'll briefly touch on the different types of collagen, not for you to memorize, but rather just to appreciate that the different types of collagen exist, that they're located in different tissues, and that we can infer a few things about them. So does anyone remember any key features of the glycine amino acid? Yeah. Yeah. And it being benign, what does that tell us about its hydrophobicity? Is it hydrophilic or hydrophobic? Raise your hand if you think it's hydrophobic. Correct, it's hydrophobic. And so if you look at the top right image, you can see that you can see the XY amino acids on the outside, but then there's the glycine is like the really small, which also is indicative of kind of like its size because it has such a small side chain. It provides the backbone of the collagen alpha helix. X is oftentimes proline, but like I said, it's not always. It's not exclusively glycine, proline, hydroxyproline. And as was mentioned a moment ago, that proline has a kink, and that kink provides a lot of strength and rigidity, and also is also important in the helical structure because again, the proline side chain turns back up to the amino group, and so that is important in the alpha helical structure of collagen. And Y is oftentimes hydroxyproline, and so it's called hydroxyproline because here you can see this is the structure of the proline amino acid, where the side chain here at the first carbon, the alpha carbon, kinks back up, but it's called hydroxyproline because it's hydroxylated here in the rough ER. That's prior, and that's oftentimes prior to the procollagen assembly. And this hydroxylation is really important because collagen is the only hydroxyproline is unique to collagen. It is so unique to collagen that oftentimes when we're looking at collagen content in organs, either from mouse or from tissue samples from patients, you can break down that tissue or that organ all the way down to its constitutive amino acid components and use hydroxyproline content as a surrogate for seeing how much collagen is there. And so kind of like the gold standard, it's not the best, but the gold standard right now for trying to figure out collagen content in tissue is doing a hydroxyproline assay to see how much hydroxyproline is present and using that as an indicator of how much collagen there is. And so hydroxyproline is only present in collagen, and it's the hydroxylation of proline there. And so here we have a table that that is meant to illustrate the diversity of collagen. And so we have fibrillar type collagen, collagens 1 through 5. As we discussed last time, we have sheet-like networks of collagen type 4. And there's also fibril associated collagens. So collagen type 9 is oftentimes affiliated with branching or nucleation from existing collagen fibers. And what's also interesting is you look down to the more recently discovered collagens, type 27 and type 28. Type 27 is considered transmembrane protein or transmembrane collagen. So that means that when it was discovered, there's oftentimes orphan proteins or orphan receptors that people are still discovering in cells. And so when this was discovered not too long ago, they're able to see, because we talked about this in the second lecture, they were able to see a hydropathy plot when looking at the amino acid sequence of this protein and see that, okay, there's this many amino acids that are hydrophobic, so that means it's transmembrane protein. And not only that, it has something else about it. What are the two key features that we talked about that are distinguishing or unique to collagen? The two key features. Does anyone remember? It was from a few slides ago. What's the amino acid sequence that's unique to collagen? Yeah. Yeah, or GlyXY. Yeah. Oftentimes, yeah. And so they're able to see in this transmembrane protein that it was GlyXY throughout the entire protein, and they're able to designate that as a new type of collagen. Everyone in science wants to discover something new, a new cell, a new protein, and so these folks are able to discover a new collagen subtype. Are there any questions so far? Mm-hmm. Of what? Sorry, I didn't hear that last part. Yeah, so with collagen, I'll actually advance to the next slide because I'm going to talk about the structural hierarchy. For collagen, it has these going from its basic to more complex structure. we have the triple helix that makes up a single collagen molecule. And collagen will oftentimes, there's pro-collagen when it's made, and the caps will be clipped at both ends, the N terminus and C terminus. And what will end up happening is that collagen will want to first polymerize associated with other collagen molecules from end to end. So you can basically imagine like it's, this is a quick snapshot into my world, like a little kid train set where like you're building the tracks and they're just like end to end, end to end, end to end, end to end. But then after that, so this is at the single molecule level. These are single collagen molecules that are attaching from end to end. The next level of complexity is these collagen fibrils that then emerge because collagen what end-to-end assembly will want to become laterally associated. And so if you imagine what provides the greatest strength to a rope, if you have a single strand of rope as opposed to like a triple strand of rope, these are laterally associated, just similar to that. And so you have laterally associated collagen fibrils that then assemble together to create a collagen fiber. Okay, so we go from the triple helix, that's 3 alpha chains together, the triple helix, to the collagen molecule level that then come together to associate together laterally to create collagen fibrils. And the fibrils are then bundled together to create a single collagen fiber. Are there any questions there? Yeah. So repeat that again. How do the fibers become fibers? It's oftentimes similar to it's oftentimes similar to the fibril assembly that they again will laterally associate to form these fibrils, or to form these fibers. Sorry. What's acting on that? So oftentimes it's also mediated by cells. So the integrin receptors on cells will also oftentimes assemble these collagen fibers together. So if you think about the not only the structural hierarchy, but also the size of each of these, a collagen fiber is several microns in diameter. Okay, does anyone remember how big a cell typically is? 10 microns, yeah. And oftentimes the cells that are doing this are fibroblasts, which, based on my experience, are usually on the order of 30 microns. They'll oftentimes change their size depending on, not to anthropomorphize them, but oftentimes whether they're like how angry they are. And so typically like angry, more scar-inducing fibroblasts will get larger because their cytoskeleton is, their actin cytoskeleton is getting ready to grab onto and become contractile. And that contractility is pulling collagen, they're spitting out collagen and assembling them together to create more scar tissue. So it's cells that are coming together and assembling these collagen molecules into fibrils, into fibers. Does that make sense? Yeah. So oftentimes the organization of these fibrils are tissue specific. If you think about the different parts of our body that move in different ways or that experience force in different ways, the ECM present there is organized to meet the demands or meet the needs of that organ or tissue. So if you consider, so we have tadpole skin that's present there. These are, if you consider it the z-axis coming out of the screen here, you have them organized perpendicularly to each other in sheets. And so that's meant to meet the demands there. If you think about how our tendons move, or our tendons and ligaments, oftentimes they're uniaxial movement. If you think of the tendons that are present, that are attached to your bicep or other muscles, but just using the bicep as an example, like it's only moving in one direction typically. And so the collagen fibers are oriented in a single axis direction to help provide tension and strength in that one single direction. Can you guys think of other organs or tissues that might require their ECM in specific orientations? Another example would be our blood vessels. Our blood vessels, while there might be some axial movement this way, but they're oftentimes experiencing circumferential stretching. And so oftentimes the ECM around our large vessels have their ECM, the collagen fibers wrapped around circumferentially to meet the demands of what the tension and the stress that that site experiences. So we're talking about fibers, we're talking about to be really reductive, like protein strings, basically. These things, if they're experiencing tension, are gonna be sliding past each other. So how are they stabilized? They're often stabilized by forming cross-links between collagen fibers. And that's typically how we define mature collagen fiber networks. And that's mediated by something called lysyl oxidase. So using some of the things we've learned so far, what's something that you think you can tell me about lysyl oxidase? Yes, it's an enzyme. What do you think this enzyme does? Just looking at the name. I'm hearing whispers of the right answer. Yes, it mediates oxidation. Does anyone want to take a stab at, without looking at what's on the slide here, if it's lysyl oxidase, what is it oxidizing? Very good. Even if you read the slide and just said it out loud, I appreciate that. So lysyl oxidase, it is catalyzing oxidation on lysines. And what that's going to create is it's going to create aldehydes from those lysines. Aldehydes are incredibly reactive, and they'll undergo spontaneous reactions with either other aldehydes or other unmodified lysines to form crosslinks there. And those crosslinks stabilize the collagen fibers within the ECM. So we've talked about how proteoglycans and their associated GAG chains can help provide strength in response to compression. We've talked about how collagen fibers can help provide strength in response to tension. But especially in response to tension, after that strain has happened, it's really important for it to be able to go back to its original conformation or to its original state, because if, for example, we're constantly feeling we constantly have the weight of our skin or the weight of gravity on our skin, it's important for our skin to be able to retain its structure, or if we're constantly pulling on our skin, the ability for it to go back. And that is mediated by elastic fibers. A key component of that is elastin. So elastin, this is really important to remember, is highly hydrophobic. I want you to keep that in mind. It's highly hydrophobic. A common amino acid repeat is VPG-XG. And so another important thing to keep note is that it does not have any glycosylation or hydroxylysines. And it's oftentimes made in its soluble precursor form, tropoelastin. And so when it's modified into its more mature form, it becomes insoluble. And so when the cells that are making or responsible for making elastin, it's first secreted in its pro-protein form, tropoelastin. And it undergoes cross-linking pretty similar to collagen by either lysyl oxidase or other enzymes of the lysyl oxidase family. And so there are different segments of elastin proteins. There is a hydrophobic part that is really important for the elastic properties of elastin. And there are alpha helical segments that are lysine rich, which is where the cross-linking occurs. And so this is, the elastin is really important in, as I described earlier, in arteries where collagen will provide the strength in response to a bolus of blood after a heartbeat. But again, it's really important for it to go back to its original structure. So can you guys think of other organs that undergo strain or undergo some sort of, that are strained and go back to their original size? Yes. Yes. That's really important because if we didn't have elastin in our lungs, we would take one breath and that's it. Another one. Yes. Yes, that's really important because if I didn't have elastin, by the time I'm 5 years old, my skin's probably just gonna be hanging down like crazy. Is there another example? Yeah.
Blood vessels.
Yep, exactly right. Another one. Yes, that's a great one. That's really important. Yeah. Yeah, exactly right. Anyone else? Anything else that probably, sorry? Muscles, it's not exactly the same because oftentimes what's happening is there's collagen breakdown that then is reassembled. But no, that's a fair one. Yes. Sorry? Your blood. Yes, that's a really important one. Another one is the heart. The heart is rich in elastin as well. And so everyone's answers kind of helps demonstrate that elastin is incredibly important. It's oftentimes easy as someone who's kind of biased. I only think about collagen because I work in the field of scar tissue and scar formation. So I only think about collagen, but elastin is incredibly important. And we're going to talk about why its hydrophobic properties are so important. Looking at the structure of elastin, as I mentioned, it's incredibly hydrophobic. So if you guys remember, things that are hydrophobic are typically embedded and hidden because our proteins and cells are usually in a very aqueous environment. And so what ends up happening is that you have our elastin. So there's elastin is the protein. We have elastic fibers. Elastic fibers are made up with a core that is elastin, but it has these microfibrils on the outside that are made of fibrillin. And so what ends up happening is that without stretch, in the image to the bottom left here, in the absence of stretch, we have elastic fibers that are cross-linked together that are bundled up. Because again, elastin is very hydrophobic. When there is stretch, what ends up happening is that these very, very hydrophobic regions are exposed. And so what ends up happening is that, as you can imagine, after the stretch is, after the source of that strain or stretching is released, elastin is going to want to bundle it up right away and hide that hydrophobic region. And so the combination of GAGs and elastic fibers help provide this balance of proteoglycans are really hydrophilic and they want water, and elastic fibers are very hydrophobic. And so these two together provide a really important balance in being able to provide resistance to compression and to provide the ability to return back and relax after the source of stretch or strain is gone. Are there any, before I move on, are there any other questions there? I promise you it's going to get very interesting very soon. Yes. Did they ever get what? Sorry. That's a good question. I'm not really sure. I mean, typically that's usually not a problem because they're oftentimes cross-linked together. So like in a way, like they're a mature elastin set of elastic fibers are kind of caught up together in a way already. And so that typically happens. And I think that's just kind of typically how elastin networks are. That's a good question.
So it's never a point where it like decreases
the ability of the relaxation. Typically, what hinders the ability, the efficiency of relaxation is actually the loss of elastin. So throughout aging, I was going to say throughout the aging process, as we age, we lose elastin, which is part of the reason why wrinkles form, which is part of the reason why, for lack of a better phrase, skin sags. For lack of a better, and so oftentimes the idea of them getting caught up on one another to hinder relaxation is typically not an issue, but rather it's the loss of elastin over time as we age that hinders the ability of things like our skin to go back and relax. Are there any other questions? Yes.
The microfibers that can help elastin, those are glycoproteins, but the elastin itself is just a normal protein, doesn't have any.
Correct, but it is cross-linked. The elastin fibers are cross-linked together. Yeah. So the last ECM protein is fibronectin. Now, what's important to know is that fibronectin does not provide very much mechanical integrity to our ECM, but it is incredibly important as a signaling hub, as a biological hub for the ECM. We'll talk about in a moment how it will provide signaling cues to different cells and alter different ECM proteins around them. And just to warn you, this is also what my postdoc advisor works on, so I've been indoctrinated to think it's incredibly important. It's the center of the universe. So fibronectin mediates a wide variety of processes. And so to the left, we have a single fibronectin protein stretched out that also has annotation describing what different parts of it do. There are three primary domains or three different types of fibronectin proteins. We have, as you can see from the bottom here, we have fibronectin type 1, type 2, and type 3 units. And starting at the very top, we have regions of fibronectin that can bind to heparin, which again, heparin is a growth factor binding glycosaminoglycan. And so this is a part of fibronectin that can actually serve as a site for growth factors to bind and hold on to and be a depot site for growth factors on fibronectin. It can also bind to collagen, but beyond that, it can also bind to many different kinds of integrins. As we talked about last class, integrins are the receptors for the ECM. And so typically we have, is it on this slide? No, it's on the next slide, forgive me. In the next slide, I'll show this, but there are lots of different integrins that usually bind to specific ECM proteins. Fibronectin is pretty unique. It can bind to a lot of different integrins, and different integrins induce different behaviors from cells. And so fibronectin, while not important for mechanical integrity, is a really important signaling scaffold protein in the ECM. So integrins, as I mentioned a moment ago, and as I mentioned before, integrins are the receptors for the ECM that cells have. And so we have typically 2 units that make up an integrin receptor for ECM. We have an alpha unit and a beta subunit. We have 18 alpha units and 8 beta units. And the different combinations that we have or that have been recognized lead to 24 different types of integrins. And so usually there is an intracellular domain and an extracellular domain. As you can imagine, the extracellular domain binds to the ECM proteins. But what's really important to note is that the short tail on the intracellular part here is actually oftentimes they become active, and after they bind to the ECM, it's going to trigger this assembly of so many proteins coming together that will actually bind to the actin cytoskeleton, onto an actin filament. And oftentimes what happens when our integrins, for example, bind to the ECM. And if they're sent, they're oftentimes kind of like interrogating around them. So you can think of integrins as like the hands and feet of cells to kind of like sense what's going on around them. And they'll oftentimes grab onto something. And if it's really, really stiff, so you can think of one of the things that we do in our lab, in the lab I'm in, is we understand like how cells sense stiffness. We oftentimes think of only cells kind of responding to growth factors, responding to proteins, responding to maybe changes in pH or temperature. They'll actually sense stiffness. So these integrins will bind onto their collagen ECM, and then they're going to slowly start to match their stiffness to the outer stiffness, because the integrins interacting with the outside area are actually connected downstream to their actin cytoskeleton inside. So what they'll actually start doing is more and more integrins will come in, and they'll start trying to strengthen that bond there. And it's going to start creating more actin cytoskeleton filaments to polymerize, and you'll get a huge giant actin cytoskeleton that looks like all these crazy fibers going across, because the cell is pulling and yanking on their surrounding matrix to try and match the stiffness. And so, like, one of the things we do in our lab is trying to figure out how do cells, when scar formation starts to happen in the lung, before scar formation starts happening in the lung, there are cells that start to think, there's something making me want to create a scar, so I'm going to go ahead and start doing it. And what they'll do is they'll start making down more matrix, and then other cells will start sensing around them. They're going to start pulling on the ECM and interrogating and realize, okay, I need to make more. I'm super activated. I'm going to make even more ECM now. And so the take-home message I'm trying to say right now, just because I do this all the time, I think this stuff is really cool, is that the integrins have an extracellular part and intracellular part. The extracellular part binds to the ECM proteins, to different versions of them, depending on the alpha unit and beta subunit. And intracellular parts will actually have lots of different proteins like vinculin and talin coming together that then are also connected to the actin cytoskeleton. And so this is a diagram that's just meant to try and illustrate how different subunits of integrins will recognize or bind to specific ECM ligands. I'm not asking you to memorize this because this is not really laid out in a way that's easy for you to memorize, but rather it's just meant to try and illustrate that there are some that only bind to laminin, like alpha-3 beta-1 or alpha-6 beta-4, some that bind to RGD receptors. So RGD is the sequence in fibronectin and collagen that some integrins are just meant to recognize. They see RGD and they say, I'm made to bind to that, and they will bind to it. And so oftentimes that's, for example, we study alpha V beta 3 a lot in our lab. We also study alpha 5 beta 1. And so those are RGD integrins that recognize that. It's called G-Fogger. And these what's considered collagen receptors, alpha 1 beta 1, alpha 2 beta 1, and also immune cell integrins that help immune cells home to the site of, if there's like, oftentimes when we have swelling or some sort of infection, immune cells will go to that site because our blood vessels have a specific protein that goes up. And these are the integrins our immune cells are able to find them, catch them, and go into the site. Are there any questions there? Yeah. Yeah, so the mechanical support yeah, so that's basically this is not important for this class, but the mechanical support is oftentimes them sensing I say sensing, I'm trying to be super reductive. So the process is called mechanotransduction. Mechanotransduction is the means by which integrins will sense the stiffness of their surrounding area, and they will in turn prompt the release of more collagen and provide more stability. And so like this is really good during wound healing because if you get a cut, you need collagen to try and provide structural integrity to that site there. And so integrins will bind and help pull things together. In addition to, as we talked before, epithelial cells sensing the changes in laminin and migrating across. But fibroblasts will also use their integrins to sense that there's something different here, and that then respond and express collagen and recover the mechanical integrity of that location. And so what's really unique about integrins, oftentimes when we talk about receptors, we talk about they will bind to some protein, some growth factor, and that will elicit some downstream consequences. Integrins are really, really unique because they can basically signal both ways. It's not many receptors will do that. So this is one way in which integrins are really unique. And what I mean by that is the outside-in signaling is they'll bind to their ECM proteins, and that will prompt downstream signaling cascades that, as I was describing a moment ago, can prompt potentially more collagen secretion. But also, interestingly, there can be some sort of other signaling event that occurs that then prompts the expression of more integrin to the surface that then will bind to their ECM or will prompt changes in existing integrins at the surface. One thing that can, let me see if I'm going to scoop myself here. That's on the next slide, so I won't say that. And so this is the outside in or inside out signaling that's really unique to integrins. So an example of outside in, I think I just scooped my entire slide. I just went ahead and did that. But an example here of outside in signaling is oftentimes integrins, before they bind to their ECM ligand, they are in a bent and what we call inactive conformation. But when an integrin will, when it sees and when it recognizes its specific ligand that it's made for, so for example, if this is alpha-1 beta-1 and there's collagen there, well then, as you can imagine, it will bind to collagen, and that collagen will have a specific charge that then will elicit a conformational change. I'm going to begin, at some point, I'm just going to start saying that something binds to something, and because it's a protein, it elicits a conformational charge. So when I do this, that's going to be conformational charge. Please don't leave me hanging like this. So it'll elicit a conformational charge, and it'll go from a bent and an active conformation to an extended and active conformation. But what's important to know is that when integrins in active conformation, the intracellular components are close together. When it's active, separate, and upon their separation, what will begin to happen is that that separation will now leave space open for those other proteins to come in. And so vinculin and talin will come in and bind to the beta subunit, and subsequently actin cytoskeleton, the actin filaments will polymerize there, and you'll get an anchorage between the outside to integrin to the inside with the actin cytoskeleton. And one thing, I don't think I mentioned this during that cytoskeleton lecture, but oftentimes the presence of, or the going from G actin to F actin, having more actin fibers forming, will actually release other proteins that will bind to our DNA and basically turn on genes or transcribe them. And so like this is one really cool way where integrins binding to an ECM will have downstream consequences that will turn genes on. And so that's outside in activation. Inside out is talin will actually compete when there's already open integrin. Talin is trying to compete with the alpha chain over to the beta chain binding site. And so this competition with talin will actually promote linkaging of integrin to become available and bind to the outside ECM. And so this will drive the integrins into an active conformation. And so if you overexpress talin, like if in a experimental setup, if you overexpress talin in a cell, you'll see throughout it, you'll see extended integrin conformation. So here, this is a I think this is a crystallography image of integrin in an inactive and active conformation. And what they did here was they just threw in a bunch of RGD peptide in this case, and that opened it up into an active conformation. So what I'm trying to describe here of inside out, inside, if you increase talin, you can actually promote the active conformation of integrins to allow them to bind to their nearby ECM. Are there any questions there? So the important takeaways that I want you to note here is that integrins have a lot of diversity. There are 24 different combinations of their alpha units and beta subunits. They bind to the local ECM, and they participate in inside out and outside in activation. The point here is not for you to try and memorize the exact dynamic of vinculin and talin here or mechanotransduction, but rather to get you to appreciate that these integrins are really important in being able to respond to ECM or respond to other cues that alter their ability to interact with the ECM. So if you could imagine our cells cannot constantly be making ECM, because then we wouldn't just be 25% collagen, we'd be 100% collagen. There has to be a regulation of ECM protein synthesis, and that's ECM degradation by proteolysis. And so we have a couple key enzymes that are really important here. So we have one class called matrix metalloproteases. Would anyone like to describe what they think a matrix metalloprotease, what's important to what we can glean from the name matrix metalloprotease? Yeah. Mm-hmm. It's exactly right. So it's an enzyme that targets the matrix, and the metallo part tells us that it requires a metal ion. The metal ion is the cofactor. So if we remember from previous lectures, a cofactor is some sort of molecule that helps provide the activity for an enzyme. Without it, it otherwise wouldn't be very active. And so in this case, it's for metalloproteases, it's a metal ion. Another class are serine proteases, plasmin and thrombin. So these are really important for clotting. You might recognize thrombin from the clotting cascade, but plasmin helps to mitigate the cleavage of, it helps to dampen the effect of thrombin to help balance it out. And so if you can imagine in a wound, again, if you get a cut, the fibrin clot, it's kind of like this is a current area of research. The fibrin clot is like an early ECM matrix that kind of helps. It doesn't just stop bleeding, but it's like an early matrix that helps kind of, we call it the provisional matrix. It is rich with a lot of cues from fibrin, and fibronectin is present there too. And then later on, after plasmin and after the provisional matrix has gone away, we get a fibronectin collagen-rich ECM protein matrix. And so while it might seem like the roles of plasmin and thrombin are just kind of relegated to just clotting, the fibrin clot is also really important. This is a new area of research. It's really important in wound healing. But for what you need to know here, matrix proteases are specific to the matrix. Plasmin and thrombin are specific to the fibrin clot during clotting. And so what they'll do is they'll oftentimes break down the matrix. So looking at the figure to the bottom left, you'll get matrix metalloproteases that will break apart the existing matrix. It'll expose the hidden growth factors as well that are within the ECM, and it'll expose cryptic sites. And so after those cryptic sites, and we talked about this with laminin last lecture, those exposed cryptics are sites that are originally hidden, but then when exposed will elicit a specific process or event. We'll have cell migration and cell adhesion. And again, matrix metalloproteases are also able to take growth factors and re-embed them within the new matrix. And so to the table to the right, we have a list of different matrix metalloproteases with their other pseudonyms, as well as their specific substrates that they target. So for example, MMP1 is also called collagenase. And as you can imagine, or sorry, collagenase 1, because we have different types of collagenase, it targets different kinds of collagen. And so we have, how are these enzymes then regulated? We have what's called local activation. And so this is pretty similar to what we described when collagen has in its pro-collagen form. At the cell surface, there are enzymes that will specifically clip and release the mature form of collagen. And so the local activation is a really important, it plays an important role for activating precursors that are being secreted. And sometimes will require a co-substrate like matrix metalloproteases that require a metal ion. Another form is where it's confined to the cell surface receptor. That's basically what I was just describing a moment ago with some MMPs that are bound to the surface, but then also there are others that are bound to a specific part of the surface of a cell that help mediate, that are important with cell migration. But another form of regulation are inhibitors. So we have tissue inhibitors of metalloproteases, we call those TIMPs. And there's also serpins that play a role in inhibiting the serine proteases that we talked about previously. So taking this all the way back to regenerative medicine and tissue engineering, how is all of this used in regenerative medicine and tissue engineering? One way that this is used is the ECMs will have tissues that's been collected and will totally decellularize that organ or that piece of tissue. That means taking away every single cellular component and but you retain the proteins, you retain the ECM that has not only the structure, but the cues that are oftentimes present. This was my capstone project, however many years ago now, which is scary to think about. But that's one way in which people in tissue engineering and regenerative medicine have leveraged the ECM. They'll have a decellularized ECM product, and some of them are trying to get to market, but there's also, again, biological problems, because if you're taking a decellularized heart from a pig and wanting to use the valve as a valve replacement in people, there is a lot of concerns if you're going to have if there's an immunogenic problem there. So another approach that people have done is to try and create engineered biomaterials that mimic the ECM. This was my PhD project. And so a lot of times people will try and make polymer biomaterials or 3D printing something, depending on the resolution of the 3D printer, that will have this, not only have the architecture of the ECM, but also sometimes will include specific proteins or lipids or gag chains that are meant to mimic the signaling cues. But what we're starting to appreciate now is that what's really interesting is that sometimes the general structure of some of these materials are actually sufficient to change responses. There's been a lot of work showing that if you have some sort of biomimetic biomaterial, if it has certain pore sizes, that will either promote angiogenesis and tissue integration or promote rejection. And so that's a really emerging area within tissue engineering and regenerative medicine right now. So I'm just going to take I'm going to take about 5 minutes to describe some of the work that Tom Barker does as a way to show how tissue engineering and regenerative medicine uses the ECM here at UVA. Again, I've been indoctrinated by him to think this is really cool and this is the center of the universe. If this doesn't interest you, and that's okay if it does, because there's so many different areas of research, I'm going to have a slide at the end that recognizes a few people at UVA, but also a lot of people all over the country, because there's also opportunities for you to go to their universities and do summer research in their lab and get paid for it. So Tom cut his teeth working on fibronectin and trying to see how the different growth factor binding sites can be regulated in wound healing. But more recently, he's kind of changed he's kind of changed a lot of people's opinion on fibronectin, because what he's noticed is that, when things are strained, we talk about cryptic sites. Cryptic sites can also be exposed in response to force and strain. And so there's an RGD binding motif right here, RGD on fibronectin right here. But there are cryptic sites that also become exposed when fibronectin is stretched. And what Tom has demonstrated is that on the order of angstroms, that's 10 to the minus 10. When there's a stretch in fibronectin, when it goes from a 35 angstrom distance down to 85, what ends up happening is that there's a site that becomes available that was otherwise not available, that then totally changes how fibronectin is recognized. And this is what one person's PhD was that just graduated this year. Those sites can be oxidized. And what ends up happening is that when those sites are oxidized, they can't go back. They stay in that strain conformation. So what ends up happening is that in some way, this is a term that Tom liked to coin, even if you no longer have that strain, it's oxidized, and there are things in here that are keeping it in this strain conformation regardless of what's going on around it. In a way, the matrix here on fibronectin has remembered that event that strained it. And so now, even if, for example, whenever people have, whether they taking this to the lung, if people are smokers, if people have exposure to asbestos, if people have exposure, overexposure to silica, there's like some sort of event that occurs that causes strain. This site is oxidized and it doesn't go back, it stays in that way. In the way it event. And cells begin to see this, they think fibronectin is strained, they think scar formation is occurring, even though nothing's going on anymore. It remembers what happened, and now cells are going to start beginning to express a scar-like behavior. And so one thing that was really cool that Tom did was he used the fact that the glycine amino acid that all of you are very, very familiar with is very benign, that has a very basic side chain. He decided, can I insert 4 glycines here to create that same separation that we're going to call 4G, because he put in 4 glycines. Now, can we use this to study what is happening? What are the exact molecular events that are happening where cells are either sensing and seeing that there's a stretch or that there's no stretch. So 4G is stretch, 9-star-10 is no stretch. And what we saw was that specific integrins recognize stretched fibronectin and specific integrins recognize non-stretched fibronectin. And the ones that recognize stretched fibronectin are really associated with scar-forming fibroblasts. And so one of our collaborators wanted to take this and try and see what are the, how can we see this at the cellular level? And so, like I mentioned before, mechanotransduction, cells sense their surrounding area. So if you put cells on glass, so our tissues are usually like, for a fibroblast in a lung, it sees about a 5 kilopascal environment. Glass is on the order of It's 3 times 10 to the 7 kilopascal. So it's 10 million times stronger than or stiffer than our native tissue. So that's super physiological. Our cells typically don't see that. And what they'll end up doing is they're acting inside a skeleton, very different from here on a 2 kilopascal. What we normally see is it becomes super strained and contractile. And so we wanted to figure out, okay, regardless of the substrate, if you put it on a surface that has 9*10 or 4G, the strained versus unstrained, on different stiffnesses, where this is like healthy lung and really scarred unhealthy lung, what happens? So here, over time, you see more actin cytoskeleton stretched, and you see alpha SMA is a marker of scar-like fibroblasts. You don't see very much because it's not there. It's the fibronectin is stuck together in a not strained conformation. For 4G, doesn't matter stiffness. You see really robust stress fibers, and you'll see this marker for scar-like fibroblasts present regardless of the stiffness because it has 4G fibronectin. And so this is where our collaborators jumped in and said, this integrin specificity, how does this exist in angiogenesis? What they're able to see, I'll make this my last slide of science stuff because I can just keep going. If you knock out integrin beta 1, our blood vessels become incredibly leaky. So in the green is a marker for blood vessels, and the red is TER119, which is a red blood cell marker. So that's supposed to be like where you should see, if you see that around, that's blood leaking out because that's red blood cells. If you knock out integrin beta 1, our blood vessels become incredibly leaky. It just moved on without me, forgive me. And that demonstrates that vascular integrity is dependent on integrin beta 1. So there are lots of different ways that you could look at this, but there is an incredible group of people across the country that use the ECM for regenerative medicine and tissue engineering. My postdoc advisor is Tom Barker. Don Griffin uses incredible biomaterials that leverage being able to mimic the structure of the ECM and the porosity of it to promote angiogenesis and tissue engineering or tissue regeneration. Aside from them, my undergrad advisor, Bo Choi, who went to Georgia Tech, who's now in Minnesota, has been doing this for a long time, and his advisor is a guy named Cato Lorenzo, who is at UVA. Few people have received the designation of university professor at UVA. He was one of them. He's now the dean of medicine at UConn. And while he was getting his MD at Harvard, decided on the side, I'm gonna get a PhD from MIT with Bob Langer, the guy that discovered tissue engineering. So like, he's honestly one of the most brilliant people I've ever met. This is the professor, her name is Tatiana Segura at Duke. She worked with Don Griffin to create their really cool biomaterial system that promotes tissue engineering, regenerative medicine. And she's the one that did that vascular integrity thing with beta 1 knockouts. And lastly, Garcia, Georgia Tech, does this to a degree that few people have done. He also leads the bioengineering unit at Georgia Tech. And Paula Hammond is the chair at MIT in chemical engineering. She does really cool nanoparticle stuff for tissue engineering and regenerative medicine that has different ECM components that I could talk about this all day. That's really cool. These are all really great people that do amazing work. I encourage you to look at them or look them up. And thanks for giving me your time. I went over 2 minutes, forgive me, but y'all have a good day.